Dear colleagues and friends, this is Mingyue Gou from Henan Agricultural University in China. It's my great pleasure to share my work here at the ACPSB-2026 meeting. My topic is Deciphering Mechanisms Underlying Maize Multiple Disease Resistance while Disease Niche Mimic Mutants. There have been increasing pandemics of multiple maize diseases in the major maize production areas in China and also in the world, including various fungal, stalk, and ear diseases. For instance, the southern corn rust and stalk rot occurred in Henan province of China in 2021, causing severe reduction of maize yield. Therefore, studies on mechanisms underlying maize disease resistance and breeding maize varieties with enhanced disease resistance is critical for maize production. In the past decades, increasing attentions have been paid to the identification of maize disease resistance genes. Other than many QTLs identified, more than 30 genes have been cloned and verified in maize, constituting valuable gene pools for improvement of maize disease resistance. Among those resistance genes, a few have been implicated in resistance to multiple maize diseases, termed broad-spectrum disease resistance genes. Those genes are involved in activation of hypersensitive response, phytohormone biosynthesis, lignin biosynthesis, phytonexin production, and antifungal protein production. However, a few puzzles remain to be solved. For instance, there are 3 types of pathogens: nicotropes, biotropes, and hemibiotrops. How do maize simultaneously defend against the different types of pathogens? And how do maize plants balance growth and defense? To answer these questions, maize mutants with growth and disease resistance characteristics are required. Plant niche mimic mutants are mutants that show spontaneous lesions without pathogen infection. Those mutants have been found in many plant species, including Arabidopsis, maize, rice, and wheat. Recent studies of niche mimic mutants in rice and wheat indicate them as valuable resources for disease resistance studies in crops. In maize, niche mimic mutants have been identified and studied since last century. It was estimated that at least 200 niche mimic gene loci exist in maize genome. However, only a small number of genes have been cloned, including LLS1, NES22, RP1-21, and ZMMM1. I started working on niche mimic mutants in Arabidopsis about 20 years ago, and my group now uses maize niche mimic mutants as a model to study maize disease resistance. We connected a series of niche mimic mutants and systemically characterized those mutants by phenotyping, DAB staining, and pathogen test, and mutants with different phenotypic and resistance types well studied. These niche mimic mutants are classified into several groups by different lesion size and characteristics, including the big lesion, medium lesion, small lesion, chlorotic lesion, and frequent lesion mutants. For instance, the well-known embryonine MO17 has typical frequent lesion. We also classify maize niche mimic mutants according to their resistance to diseases caused by different types of pathogens. For instance, NES8 is resistant to the southern corn leaf blight caused by nicotropic pathogen. NES30 is resistant to coronarial leaf spot caused by hemibiotropic pathogen. NES33 is resistant to the southern corn rust caused by biotropic pathogen. We used 3 types of niche mimic mutants, NES4, 10, and 17, to dissect the general transcriptional changes. We found that defense-related pathways, for instance, programmed cell death, defense response to fungi, phenylpropanoid biosynthesis, were commonly activated in all three mutants. We propose that mutation of those niche-mimic genes could lead to upregulation of defense-related genes and downregulation of photosynthetic genes, and there appears to be an obvious growth defense trade-off in those genes, in those mutants. To identify the causal genes of those niche-mimic mutants, we generated F2 populations and performed BSA or BSR-seq, as well as marker-assisted linkage mapping. We have verified a few candidate genes. Those genes are involved in chlorophyll degradation, phosphate metabolisms, carbohydrate metabolisms, immune perception, and signaling transduction. I'd like to show data of three different types of niche-mimic mutants as examples here, including NAS30 with big lesion, NAS8 with medium-sized lesion, as NAS33 with chlorotic lesion. NAS30 was identified as a typical mutant with big lesion. When the mutant was infected with Coronaria lunata, very few fungal colonies could be found on the mutant leaves, and DAB staining indicates increased hydrogen peroxide accumulation in the mutant leaf. By BSR-seq and map-based cloning, the causal gene of NAS30 was identified as LLS1 that was previously reported to encode pheophorbide A oxidase, which is involved in chlorophyll degradation. We sought to dissect NAS30 comfort disease resistance by RNA-seq analysis. We found that defense response and signaling is amplified in NAS30 mutant plants in a lesion-level dependent manner. Genes involved in both jasmonic acid and salicylic acid biosynthesis are mostly upregulated in NAS30. In addition, genes involved in lignin and flavonoid biosynthesis are also upregulated. With above data, we propose that mutation in NAS30 interrupted the degradation of chlorophyll, resulting in the accumulation of pheophorbide A, which causes increased accumulation of hydrogen peroxide, jasmonic acid, salicylic acid, lignin, and flavonoids, thus conferring cell death and broad-spectrum disease resistance. The second mutant is NAS8, which shows medium-sized breaking lesions. Other than the lesion-mimic phenotype, NAS8 shows enhanced resistance to Coronaria leaf spot and southern corn leaf blight. By BSR-seq analysis and map-based cloning, NAS8 was mapped to a region on chromosome 9, and this locus was not reported before. Similar to NAS30, S-lignin level and JA level was increased in NAS8 mutant. However, SA level was dramatically decreased in NAS8 mutant, indicating different mechanisms underlying NAS8 comfort disease resistance. Besides, genes involved in the biosynthesis of two terpenoids, zinlaxin and caronaxin, were also mostly upregulated in NAS8, and co-incubation with leaf extracts of NAS8 significantly inhibits the growth of Coronaria lunata strain, implying that NAS8 comfort resistance to Coronaria leaf spot and southern corn leaf blight is largely due to the increased accumulation of the antifungal secondary metabolites. The third mutant is NAS33, which shows spontaneous chlorotic lesions. Other than the lesion-mimic phenotype, NAS33 show enhanced resistance to both southern corn rust and Coronaria leaf spot. By BSR-seq analysis and map-based cloning, we found that the MFO2, which encodes a ubiquitin conjugating E2 enzyme, is a causal gene. FO2 was previously known to mediate the degradation of phosphate transporter protein and acts as a negative regulator of phosphate homeostasis. Consistent with the known function of the MFO2, phosphorus accumulation is indeed increased in NAS33, and exogenous application of phosphate enhances the lesion formation in NAS33. Based on transcriptomics data, we detected significant upregulation of defense-related genes and downregulation of photosynthesis-related genes. To examine if phosphate could induce Maize defense response, we treated Maize seedlings with different concentration of phosphate in a hydroponic system. By RNA-seq analysis, expression of defense-related genes were activated by phosphate application in a phosphate level-dependent manner. Among them, genes involved in phenylalanine-derived cyanidinic acid, lignin, and flavonoid biosynthesis were mostly activated in NAS33. Consistently, the accumulation of SA and S-lignin, but not JA, are increased in NAS33. We asked if phosphate application could enhance Maize disease resistance by inoculation with Puccinia polysaccharide strain that causes southern corn rust in B73 maize plants. We noticed an obvious increase of resistance with the increase of phosphate level in the same hydroponic system. For the coronary leaf spot resistance of B73, we noticed a similar phenomenon. This data suggests that phosphate is positively involved in basal defense against multiple pathogens. To examine if phosphate could affect basal defense, especially PAM-triggered immunity, we performed FRAC22-triggered ROS production and MAPK phosphorylation assay. We detected an increased induction of ROS burst and MAPK phosphorylation by FRAC22 treatment in the NAS33 and ZIM42 mutants. Besides, when B73 plants were treated with different phosphate, we observed an increase of FRAC22-triggered ROS burst and MAPK phosphorylation. This data indicates that phosphate positively regulates PAM-triggered immunity. To verify if phosphate could affect field-level disease resistance, we applied the phosphate fertilizer to the field when plants were 50 days old. The southern corn rust resistance was significantly decreased when phosphate was applied at a dosage of 45 or 90 kg per hectare, depending on the basal phosphate level of the field, highlighting the great potential of using phosphate fertilizer to control maize disease while maintaining its role as nutrient. Based on our findings, we propose a working model for phosphate-mediated regulation of plant growth and immunity in maize. In contrast to the reduced growth and immunity in no phosphate-treated plants, high phosphate treatment triggers a strong immune response, including enhanced phosphorylation of MAPKs, upregulation of defense-related genes, and increased accumulation of SA, S-lignin, flavonoids, etc. However, there appears to be an obvious growth defense trade-off when phosphate level is too low or too high. Our group have recently reported that maize growth defense trade-off is regulated at both transcriptional and post-transcriptional levels, and coordinated regulation of plant growth and defense is critical for achieving high maize yield and normal and diseased conditions. We therefore propose optimized application of phosphate fertilizer as a strategy to coordinate maize immunity and growth in maize production. This is the last slide of my talk, and the take-home messages are outlined here. First, different types of niche-mimic mutants are valuable resources to dissect maize disease resistance against the different types of pathogens. Second, optimization of plant growth and defense is vital for maize breeding and production. Third, phosphate fertilizer application could be optimized to achieve coordinately enhanced resistance and yield of maize. I'd like to thank the support of my university and government for the funding support. I also thank my group members for their dedicated contribution to this study. I appreciate a lot the kind contribution of mutant seeds from our colleagues. Thank you all for your attention.